Good morning. I am Professor Sunil Khare from Indian Institute of Technology, Delhi. Under the E-Pathshala program of UGC in the biochemistry subject, today we are going to talk about central dogma. The central dogma is very essence of the life. It tells us how the information flows in the living systems from DNA to protein. The central dogma was proposed by Francis Crick in 1958. He proposed that information starts from DNA, goes to RNA. This process is called transcription, and the RNA can pass on the information to the protein, and this process is called translation. One more assumption which was there in this central dogma hypothesis by Crick was that DNA can replicate itself. So, in toto, this was the central dogma what was proposed by Francis Crick. There are two basic assumptions in central dogma. One is the genetic material which was proposed to be the DNA should be able to replicate itself, and second, that the information should originate from the genetic material to the functional unit, that means from DNA to protein. After David Baltimore discovered the reverse transcriptase, there was a major challenge to central dogma. What David Baltimore discovered was that some of the RNA can replicate itself and they can act as template to form the DNA. This is what led to a concept of reverse transcription. The question came, how this reverse transcription can be embedded in central dogma. So, the central dogma was modified, and then now it looks like DNA replicate itself to DNA, DNA to RNA the transcription. RNA can replicate itself, RNA replication, but RNA can also reverse for the reverse transcription to DNA, but then RNA information flows to protein in form of a translation that still remains valid. So, the new central dogma or modified central dogma is DNA to RNA, RNA to protein, but DNA to DNA and DNA to RNA and RNA to DNA all are possible. In 1953, Watson and Crick proposed the structure of DNA that stated DNA should fulfill all the requirements which are anticipated by a genetic material. The material should be able to replicate itself, that means it should be able to create its own copy. Next to that was a presumption that the genetic material should also be able to pass the information. Based on above, in 1954, Gamow proposed that DNA could directly determine the sequence of protein. DNA was the component of chromosome which was present in nucleus, and protein synthesis takes place in cytoplasm. Along with this, RNA was abundant in cytoplasm. So, the direct role of DNA in protein formation could not be explained. That why abundance of RNA is there in the cytoplasm and protein synthesis is taking place in cytoplasm. The rate at which protein synthesis takes place was also unexplainable. In addition, they also found microsomes in cytoplasm which were made up of RNA and protein, and in microsome protein synthesis does not occur. In contrast to these observations, Crick got one clue for central dogma, that once information reaches to protein, it will never get back to nucleic acid. Later on, in continuation of this, Crick postulated two more hypotheses. First was sequence of hypothesis and other was protein folding hypothesis. In sequence hypothesis, Crick assumed that specificity of nucleic acid lies in the arrangement of their bases, and that sequence is unique for a particular protein. At this point of time, the concept proposed by Crick was totally informational. For further elaboration of central dogma, it was based on two different experimental observations which were done by Heinz, Frankel, Cornett and Christian and Finson. In one experiment, Cornett showed that two different strains of tobacco mosaic virus could be distinguished by difference in their protein coat, and RNA is required for correct production of these coat proteins. Whereas, in Finson's experiment, it was shown that spontaneous refolding of ribonuclease enzyme denatured in vitro. All these observations allowed Crick to understand the flow of genetic information from DNA to RNA to protein. Crick stated that the transfer could be divided into three groups. The group 1, for this transfer, evidences are available in nature and they were DNA to RNA, RNA to protein. These are four kinds of transfers under this category. The possibilities are that DNA could transfer to DNA, DNA could transfer to RNA, RNA could transfer to protein and RNA could transfer to RNA. All these transfers are evident in most of the living cells, and last one is found in RNA viruses. The group 2 proposed by Crick was, for this group transfer neither any experimental nor any strong theoretical evidence were available that time. This group 2 is DNA to RNA and RNA to protein. Transfer in this category could be RNA to DNA, for which the evidences were provided by Professor Tamin, and DNA to protein, for which the evidences were provided by Dr. Gamow. This class was probably rare and found only in RNA viruses. Group 3 consists of three transfers: protein to protein, protein to RNA and protein to DNA. This transfer was very unlikely, and for this kind of transfers, cells have to evolve totally a different kind of machinery, a kind of reverse translation, which was not anticipated. 4 rules of central dogma. So, these are 4 basic rules of central dogma. Number 1, it does not explain what kind of machinery is used for transfer of genetic material and what kind of errors incorporated during this process. It does not tell about rate at which transfer process works. Number 3, it was intended to apply only for the present day organisms and not to the past events where origin of life could have taken place. 4th, it is not the same as assumed earlier that the transfer occur from nucleic acid to protein and back. 3 kind of transfer explained by Crick. So, Crick could explain actually 3 different transfers. Number 1, he could explain general transfer. It occurs in all the cells with few exceptions, such as reticulocytes of mammalian systems. It falls in the group 1 as stated in the previous slides. Second is spatial transfer. This is not very common, and this occurs only in some of the specialized cells, such as virus infected cells, and this may fall into group 2 as discussed earlier. Number 3 are unknown transfers, which fall into group 3, and there are no evidences available for this kind of transfers. However, a spatial cell free system containing neomycin in bacterial cell can perform protein to protein transfer of information, but unusual. There have been many challenges for central dogma, and many of the research people contemporary to Crick actually opposed that, and they challenged it. Prominent among them were Howard Tamin, Satoshi Mijthani and David Baltimore. The discovery of reverse transcriptase, formation of prion, role of chaperone in protein folding, epigenetic modification of DNA, chromatin that modify gene expression, RNAi, RNA splicing and RNA editing are new information currently available, and then they supported the concept of central dogma reversal. and let us see in subsequent slides how these discoveries challenge central dogma. While David Baltimore in 1970 discovered reverse transcriptase that had a capability to synthesize DNA from RNA. That means, in such cases RNA can act as a template for DNA synthesis. However, it did not contradict central dogma, as Crick has explained. These type of transfers may occur in some special case. So, it could be considered as a special case. But after this discovery, these transfers categorized as reversal of central dogma. Second thing which challenged the reversal of central dogma was discovery of prion. After the discovery of prions, just to remind you that prions are aggregated proteins which have pathogenic capability. So, after their discovery, central dogma got affected. Prions are protein that become pathogenic after aggregation, and this is a kind of a spontaneous process. There is transfer of information from pathogenic form to non-pathogenic one, which is contradictory to Crick information and against the hypothesis that all information inside the cells originate from DNA and pass on to the proteins. Therefore, in case of prion, the central dogma does not fit very well. Discovery of chaperonins was another challenge for explanation of central dogma. In 1980s, chaperonins were discovered. Their discovery challenged the statement of Crick that protein folding is a function of the order of amino acids and no other machinery is required for protein folding. Chaperonins were known for proper folding of the proteins and present in the different compartment of the cells, which showed that Crick was totally wrong in predicting the protein folding phenomenon. However, this issue was rapidly vanished after knowing the proper function of chaperonin, that was only to prevent improper folding of protein. This again re-established that folding of protein was simply the function of order of amino acids. So, all in all we can say that central dogma still is very basis of the life, wherein information flows from DNA to RNA and RNA to protein. However, with the discoveries as discussed previously, another modifications were added, that RNA could also be a genetic material, especially in case of RNA viruses, and then RNA can be a template for synthesis of DNA. So, there is a modified central dogma now, but central dogma still persists. So, in this lecture what we have learned so far is that central dogma states that DNA flows the information to RNA and RNA flows the information to protein by the process transcription and translation respectively. The revised form of central dogma is that RNA to DNA is also possible in form of reverse transcription. This valid hypothesis of current central dogma explains the flow of information in life.